so this video is going to go into cellular respiration in just a little bit more detail. Remember, the purpose of cellular respiration is to make energy for the cells, and that energy is going to be in the form of ATP. The way it goes about doing this is by taking glucose, which is C6H12O6, and using oxygen, among other things, to convert it to ATP. Carbon dioxide and water are produced as waste products during this process. So this is the reason you breathe. You take in oxygen to use for cellular respiration, and you get rid of carbon dioxide that was generated as a byproduct of that respiration. Cellular respiration is divided into 3 stages. Stage 1 is glycolysis, and it takes place in the cytosol and does not require oxygen. Stage 2 is the citric acid cycle. It takes place in the mitochondrial matrix and does require oxygen. Stage 3 is the electron transport chain, and it takes place on the mitochondrial cristae. It also requires oxygen. This is the money step, the one that makes the most ATP out of the 3. In glycolysis, a glucose molecule is broken down into 2 pyruvate molecules. These molecules can also be called pyruvic acid, so don't let that confuse you. Your textbook uses both terms, and you'll hear me use both terms, but pyruvic acid or pyruvate, they're pretty much interchangeable for our purposes. So in this process of the glucose molecule being converted into 2 pyruvates, 2 ATP molecules are also produced, 1 for each pyruvate. Also for each molecule that's made, a hydrogen atom, along with its associated electron, is donated to NAD+, which turns NAD+ into NADH. So NAD+ is an electron transport molecule. Its job in the cell is to pick up electrons, which turns it into NADH, and then drop them off at other locations in the cell. Your textbook compares it to a taxi cab. So when the taxi cab is empty, it's called NAD+. After it picks up its electron and it's full, it's called NADH. The citric acid cycle is stage 2, and it's a rather complicated process, as you can see based off this diagram, that takes place in the matrix of the mitochondrion. It's a series of reactions that are catalyzed by 8 different enzymes that basically will break down pyruvate by first changing it into acetyl-CoA, and then harvesting its electrons, releasing carbon dioxide as a waste product. So these reactions are a cycle, because every trip around the pathway regenerates the first reactant, oxaloacetate, which is always available to react with acetyl-CoA as it enters the cycle. However, you do not have to memorize every step in this cycle, so don't freak out. Actually, what you need to know is this. So pyruvate is converted into acetyl-CoA. Acetyl-CoA then enters a series of cyclical reactions where electrons are removed to make NADH and FADH2. FADH2 is just another electron carrier molecule, very similar to NADH, and then carbon dioxide is released as waste. A small amount of ATP is made in the process, so the end products per pyruvate molecule are NADH, FADH2, 1 ATP, and carbon dioxide. Since each glucose molecule makes 2 pyruvates, the cycle actually occurs twice per glucose molecule. So that means a total of 2 ATP are made per glucose molecule. So now kind of the question is where are all these electrons going that are being carried by NADH and FADH2? Well, they're actually going to be carried to the final stage in this process, which is the electron transport chain. The ETC is a series of proteins that are embedded in the inner mitochondrial matrix, in the inner mitochondrial membrane, and these proteins serve as a type of conveyor belt, so they move electrons down the chain. At the end, the electrons and their associated hydrogen ions are going to combine with oxygen to form water as a waste product. So this movement of electrons ends up forming ATP through a very interesting process. That process is called chemiosmosis. So each time an electron moves from one protein to the next, so as it moves down the line, that protein actually transports an hydrogen ion, so an H+ ion, to one side of the membrane. against the concentration gradient. So remember we talked about active transport. This is an example of active transport. The protein is transporting that hydrogen ion against its concentration gradient. So that results in an accumulation of these hydrogen ions on one side of the membrane. So at the very end of the electron transport chain, an enzyme called ATP synthase that allows the accumulated hydrogen ions to flow back down their concentration gradient. So that movement is kind of like a water wheel, or wind turbine. It's going to provide some energy through that movement, and that energy is used to make ATP by attaching a phosphate group to an ADP molecule. So ATP synthase is able to make 26 ATP molecules from one glucose molecule. So that's why it's considered the money step of cellular respiration. It makes much more ATP than any other step in this process. Notice how important oxygen is in this step because it's the final acceptor of electrons. If you don't have oxygen, you don't have the electron transport chain. The oxygen joins up with hydrogen ions and the electrons to make water. So without oxygen, the ETC cannot occur. Without oxygen, some cells, like those muscle cells I mentioned before, can go into anaerobic respiration, which was discussed briefly in the previous video in a little bit more detail in your textbook. So I'll try to have some videos at the end of this section in Blackboard that'll kind of give you maybe some animations on how cellular respiration occurs and how the electron transport chain works and all of that. So I think once you see that in action, it'll make a lot more sense. But basically, it's a series of steps. The overall purpose of them is to make ATP. So there's a lot of kind of exchanging of electrons going on to get to the electron transport chain so that they can generate ATP to use as energy in the cell.